All right, folks, welcome to week 10 of biological anthropology, physical anthropology, whatever you want to call it. This week, you have your next lab to do. It's going to help you substantially to watch the lectures that I'm going to post, rather that I posted last week, the lecture, and then the one from this week, just to give you something to cite. Also, the reading from last week and then the POTS reading from this week should help you. You can also explore the Smithsonian Institute's website where you're going to complete the lab anyways. All right, so Hominin Evolution Lab, if you go to the document here, this is just a Word document. Part 1 is going to ask you about the Laetoli footprints. So actually, hold on one second. Let's do it this way. So part 1 is going to ask about the Laetoli footprints. These are here. Don't look up anything about the Laetoli footprints. Simply use what we've talked about in class. What is a fact, a theory? Think back to that Gregory article and use that briefly to just engage with these footprints. And so you're going to look at these scans. What are the facts? What do we know about these? So what are just the objective observations of these footprints? Then from those facts, what can we infer? So what are some of the conclusions that we can draw from those footprints? And then finally, what more information do we need to further evaluate this site? So what else is it that we need to do? What else do we need to know to draw additional conclusions? All right, so that's part 1. Part 2 is going to ask you to evaluate fossil skulls. And so I give you the species names here, mainly because when you go to it, it's going to tell you that. But you click on the link, and it's going to carry you to the Smithsonian Institute's website. And as this loads, each fossil, of which there are 4, has a column and it has a question. So we're going to answer these questions for each of the 4 fossils. So question 1, for instance, does the forehead or frontal bone look more vertical or flatter when the skull is held in the normal anatomical position? This is just the position where it's facing forward. So does the forehead, the frontal bone look more vertical or flatter? So vertical would be coming up. Think about our forehead and how it's vertical. This is right. This one slopes back. And so you're basically going to go through these questions and answer these. If you don't know a term, that's fine. You can Google it. A supraorbital brow ridge is simply the brow ridge. So this is just where your eyebrows are. So the bone, what does that bone do? Is it mostly flat or does it stick out? If it sticks out, there's a supraorbital brow ridge. Is the brow ridge divided or continuous, right? You look between here, right? This is a continuous brow ridge. It's not divided. It's one continuous bone. One thing that I wanted to point out, I guess I'll talk about each of these just to give you an idea of what they mean. So brow ridge divided, shape of the brain case, front to back. So I would look at it, you can look at it like this, but it's easier to look sideways. So is it long? Is it round? Right. So let me see if it'll let me draw. Our brain cases are globular. They're shaped like a globe. Right. This one is more elongated. Is there a sagittal crest present? A sagittal crest is going to be like so you have this line here. Let me get my drawer back out. On this skull, there's this line here. This is called a temporal line. This is where your jaw muscle attachments your jaw muscles attached to your skull. This is Homo heidelbergensis, but we have a temporal line as well. And if you go just above and behind your temples, and you clench your teeth together, you can feel that muscle flexing. You can feel right where your temporal line is. Sagittal crests are the same muscle attachment, but they're up on top of the skull. And so just to give you an idea, let's just Google gorilla and take this off. Skull. So gorillas are a great example of an animal who has a sagittal crest. the sagittal crest is this crest here. Let me draw it again. This crest here, that's your sagittal crest. This crest here is the nuchal crest. So this is a different crest. Hominins don't have nuchal crests, but some hominins do have sagittal crests. So when you're looking at each of the 4 examples, is there one of those crests present, sagittal crest? So there's a suture right here on the skull. You can kind of see it here. Zoom in a little bit. There's a suture. This is where the parietal bones meet. And then there's another suture here where the frontal bone meets the parietal bones. This suture, so there's a suture here and then a suture here. The suture here is called your coronal suture, like the sun, like a corona. Coronal suture, this is the sagittal suture. So the sagittal crest is simply the suture, the crest that runs along the sagittal suture. So is there a sagittal crest present? In the normal anatomical position, is the foramen magnum oriented downward or more towards the back? So we can look at the bottom of the skull. The foramen magnum, remember foramen just means whole, magnum just means big. So is the foramen magnum angled downward in the normal anatomical position, or is it more towards the back of the skull? And the next one, is the mastoid process relatively flat, or does it noticeably protrude? So the mastoid process is going to be the bone process right here. So you can feel your mastoid process. If you reach behind your ears, you feel there's a little protrusion of bone just behind your ear. That's the mastoid process. Some hominins have a mastoid process. If we look at a human skull, you'll notice that I'll give you an example of a more pronounced mastoid process, if I can find one from the side. Here we go, this is good enough. So this bone here, poking down, that's your mastoid process. So that's what you're looking for in that. Are the nasal bones raised or are they flat? So looking at the skull, the nasal bones are exactly what they sound like. It's the bones right here. There's 2 of them, one on either side. Let's rotate this and get rid of the drawing. Right, there's a bone here, and they meet at a suture right here between the 2. Are they raised or arched, or are they flat? Right, so right, you can see from this, they are quite arched. So now we're getting into measuring. So describe the maximum breadth of the nasal opening. So nasal opening is just this aperture right here, the hole where the nose is. So it's asking what the maximum breadth is. To access that, what you're going to do is come over here. You see the tool thing? You're going to click on that, and it's going to bring this up. Then you want to click measure, which is right here. So you're going to click measure, and then you want to turn it on. So right now it's off. So you turn it on, it turns blue. So that's not what we want. So find the maximum, the widest point of the nasal opening, and you click there, and then click the other side, and it'll give you a measurement. And anytime I'm asking you to measure anything, that's what you're going to do. So maximum height of the nasal opening. So maximum height, you're going to go from the tallest point to the lowest point in the nasal opening, right? So not in the center, because that's not the maximum. Then you're going to look at the length of the maxilla. So the maxilla, clear this off. The maxilla is this bone here and here. So it's one bone. You're going to try to get the maximum width, I believe is what it says. Let me click one more time. Look, the maximum length of the maxilla. And so from there, you're going to go from the front here. So we're going to measure from the front all the way to the back here, to the back of the maxilla. So you're going to get that length. The bizygomatic breadth. Turn the measurement off really quickly. So your zygomatic bones are going to be these bones here. They're on either side. And so what you want to do to measure that is have it facing on and see the widest point of the face. You've got zygomatic bone here and here. So that's where you're going to measure from. So you're going to measure the widest point wherever it lets you click. And then you're going to go same thing. You're going to measure that width. The shape of the dental arcade. So this is just the dentition. So what does the dentition look like? So if we look at it this way, is it U-shaped or V-shaped? Right, so is this more U-shaped or V-shaped? If you need to flip it over, you can do that too. When viewed from the side, are the incisors angled out or are they vertical? Well, we don't have incisors here. So in this case, we have to be a little bit creative. So this would be normal if you're looking from the side. Do the incisors go in this direction or are they straight up and down? Well, there's still a way to determine that. Even if we can't see the incisors, we can look at the direction of where the teeth come out. And to me, these are definitely vertical. They are not jutting out. You'll see some examples in this activity of some that are forward. Describe the combined width of the 4 incisors together. Well, yet again, we're kind of tricky because we can't actually measure the incisors. What we can do is work with what we have. And so here we want to measure. So the incisors, your incisors are your front teeth. So there would be an incisor here, an incisor here, an incisor here, and an incisor here, right? They anchor in here. So if you imagine there are incisors there, then you can measure those incisors. You can see where the bone comes down. You can see where the bone is, the tooth socket is rather, and you can then draw that in, and then you can measure across those. And that'll give you a fairly close estimate. Without the teeth, you can't be like super duper accurate, but you can get a fairly close estimate of the maximum breadth of the incisors by doing this. Does the canine tooth project above the chewing surfaces? Yet again, this is a little bit tricky if you only look at the right side of the skull. But if we look at the left side of the skull, right, so we're missing incisors, but we've got an incisor here and incisor here, which means that's the canine. Because remember, all hominins, all catarrhines are 2, 1. Actually, let me. Sorry if you hear my cat. She's being grumpy right now. So there's 1, 2. So 2, 1 canine, 1, 2 premolars, 1, 2, 3 molars. So 2, 1, 2, 3. And so we know that this is the canine. Does the canine extend below the chewing surfaces? In this case, is the canine pointy? Does it stick down farther than the chewing surfaces here? The answer is no. Just to spoil it for you a little bit. And then finally, is there a canine diastema? The diastema is just going to be, let me see, let's look at a chimp skull. So if we look at a chimp skull, that is a gorilla skull. If we look at a chimp skull here, the diastema, the canine diastema is going to be the gap between the incisors and the canine. This allows the canines to slip past one another. There's a honing complex that keeps the canine sharp, but the diastema is just that gap. So is there a diastema in heidelbergensis? Now you don't have the lower jaw, right? But you can see if you draw in or imagine where the incisors are, right? You can see that there is no gap between where the incisors would be and where the canine is. So there is no canine diastema in heidelbergensis. And so you're just going to do that for all 4 of these fossils. That'll complete part 2A and part 2B. You're going to put them in order. So you're going to have 4, the earliest one, the one that is the oldest, and then you're going to have 2 middle, and then the most recent one. So in question 1 of part 2B, you're just going to make a list 1, 2, 3, 4. In question 2 of this part, you're going to then explain to me why you put them in that order. So this is going to be looking at the lectures, using the Smithsonian Institute's website, and you are going to simply put them in, justify why you put them in the order that you did. And question 3, just what are some conclusions you can draw about each of the fossils? I'm not asking you to cite anything here. I'm simply asking you to look at the work you've done and see what conclusions you might draw based on what you know, what you've learned already. If you want to cite stuff, feel free to. But just, I want you to just think about the skulls. Measuring them isn't doing science. It's thinking about what those measurements might tell us about the organisms that tell us something about, the scientific utility of each of these fossils. Then finally, part 3. Part 3, you're going to look at Paranthropus boise. You're going to go to the website, same one as before. You're going to check out this skull, right? You've got a sagittal crest here on this hominin. You're going to look at Paranthropus boise, and you are going to compare and contrast Boise, which is part of the Paranthropines, which are a sister taxa to the Australopithecines and in genus Homo. So what are some things that you notice about Boise that is different from the other lineages that we've looked at? And then what might these adaptations tell us about their behavior or life ways? So based on what you know about evolution, based on what you know about adaptation, what conclusions might we draw from the adaptations, like having these huge, large chewing muscles, the big flat faces, the big teeth, etc. All right. If you have any questions, obviously feel free to reach out to me. Otherwise, I am really looking forward to looking through your labs once you get them all submitted. Take care and have a great week.